Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about the expression vector. Before that, will you please consider subscribing my YouTube channel? Your subscription is often a lot to me, so please do subscribe. An expression vector is usually a designed plasmid or a virus which is used for high-level expression of the transgene in the host cell. Unlike cloning vector, which only stores or replicates the target DNA, expression vector is used to introduce the transgene into the host cell where the vector uses host cell protein synthesis machinery to produce the protein encoded by the transgene. These vectors are the basic tools in biotechnology for production of different types of proteins, for example insulin, which is used for medical treatment of diabetes. Expression vectors contain certain regulatory elements such as promoters and enhancer which drives high-level transcription of the transgene. The main objective of a well-designed expression vector is the efficient production of protein. This can be achieved by promoting high-level transcription of the transgene, which results in the formation of a significant amount of stable mRNAs, which are then translated into protein. In some expression vector, for example in PT2887 LARC or GFP, the gene is expressed only when necessary in the presence of an inducer such as isopropyl beta-D-thiogalactoside or IPTG. In other cases, the gene is expressed constitutively. An expression vector contains key regulatory elements such as promoter, ribosomal binding site, transcription terminator, enhancer and silencer, and selectable marker. This figure illustrates the structure of a bacterial expression vector, which is a plasmid containing 3756 base pairs. Key components present in this vector are: number 1, origin of replication. It is an AT-rich region at which replication of the plasmid is initiated. Number 2, selectable marker, which is used for the identification of successfully transformed cells. One type of selectable marker gene is antibiotic resistant gene, which in this vector confer resistance to aminoglycoside antibiotics such as neomycin and kanamycin. The transcription of this gene is under the control of KNR promoter. Number 3, multiple cloning site. It is a specific sequence which contains restriction sites for several enzymes so that the transgene and the GFP gene can be inserted. Number 4, promoter. Promoter is a specific sequence at which RNA polymerase and transcription factor bind to drive the transcription of the gene, which is located downstream. In this vector, T7 promoter has been used. This is a strong promoter which is inducible in the presence of IPTG. T7 promoter is recognized by T7 RNA polymerase. So T7 RNA polymerase binds to this promoter and initiates the transcription of the downstream gene. Number 5, lag operator. It is a segment of DNA which acts as a genetic on-off switch, thereby controlling the transcription of the downstream gene. Number 6, lag I gene. It is another regulatory element that encodes a protein known as lag repressor. This repressor protein binds to the lag operator, thereby preventing T7 RNA polymerase from transcribing the downstream gene. But when the inducer is present, then isopropyl beta-D thiogalactoside binds to the repressor protein. This binding results in the conformational change in the repressor, as a result of which the repressor is detached from the operator, after which T7 RNA polymerase continue transcription of the gene which is located downstream. In this vector, the expression of the lag I gene is under the control of another regulatory region, that is lag I promoter. In this vector, we have inserted GFP gene and the transgene in the multiple cloning site. GFP gene encodes a protein known as green fluorescent protein, which acts as a fusion partner to track the production of protein which is encoded by the transgene. This is the transgene which has been inserted adjacent to GFP gene. So when this gene is expressed, a fusion protein is formed which consists of green fluorescent protein and the protein encoded by the transgene. Number 8, ribosomal binding site. It is a specific sequence at which the ribosome binds in order to translate the mRNA. So when the inducer is present, lag repressor is detached from lag operator. As a result, T7 RNA polymerase continue the transcription of the downstream gene. This results in the formation of stable mRNA. When the mRNA is translated, a fusion protein is produced which consists of green fluorescent protein and another protein which is encoded by the transgene. Number 9, T7 terminator. It is another key regulatory region which acts as a signal for termination of transcription of this gene. After synthesis of the fusion product, the recombinant protein which is encoded by the transgene is cleaved and purified. This is how different expression vectors function in suitable host cell for production of different types of recombinant proteins. Expression vectors have several applications such as efficient production of protein, production of therapeutic proteins, enzymes, antibodies, hormones, and cancer-treating antibodies. In addition, this vector is also used to study protein function by expressing it in specific cell type. Expression vectors are also used in genetic engineering and gene therapy and also in protein engineering. Friends, this is all about expression vector. For more updates, subscribe my YouTube channel, Sam Biology Class.